Okay, so what I'd like to talk to you today about is arbovirus surveillance in Australia, and how we've needed to pivot to JE recently in particular. And what I'd like to do to start out, before I talk more about the outbreak itself this year and what we found in preceding years leading up to this outbreak, is to tell you about Australia's arbovirus surveillance system across both medical and veterinary jurisdictions. So this table is a little busy, and I apologise for that, but it gives you, I guess, a snapshot of the different arboviruses that are important for both public and animal health in Australia. So you can see Japanese encephalitis is at the top. This is actually an old table that I've used several times before, and J has always been at the top because of its propensity to spread and cause large outbreaks, as we've seen. It's always been a major threat to Australia. Beneath that, though, we've got some, I guess, local homegrown flaviviruses closely related, Murray Valley encephalitis and West Nile Kunjun subtype. And then we have some other arboviruses, Kookaburra, Edge Hill, Septic, which are thought to be associated with disease, but there's not really a lot known about them, and they're not detected as often, not as a significant public health threat. And then we have Ross River and Barmah Forest, which are two of the major causes of arboviral disease in Australia. Both of those cause arthritis and are found across all of the states. And then in blue are the viruses that are exotic to Australia. These mostly cause traveller infections, dengue, Zika, and chikungunya. Excuse me, although we have had a number of dengue outbreaks, particularly in Queensland and Northern Queensland, that have resulted in localised transmission. So these are our targets. Now, for getting into surveillance now, there are surveillance systems for humans. Human cases, we have a national notifiable disease surveillance system, probably much like yours. This is a primary disease surveillance system, a passive surveillance system, in which it's required by law to be reported to government, both state and federal authorities, or global health agencies. Most notifications are based on laboratory definitive evidence, and the diseases on this list, most of them have laboratory case definitions that support that definitive evidence. And so on this list for arboviruses, you can see here, there's all of those viruses on the table that I showed you before. And we also have a category called Flavivirus infection unspecified, which covers Alfwe, Kookaburra, Edge Hill, Stratford, Newmapoon, and Zika viruses. So some of those rarer instances that we might see human disease. Now, on the other side, for animals, we also have a national notifiable animal disease list as well. Dr. Shipp may have talked about this yesterday in his presentation. I'll just briefly go over it. This is to cover major threats to Australian livestock industries and access to overseas export markets. Like the human diseases, these must be reported, although in this case to agricultural authorities, and this is legislated by individual states and territories. State and territories also have their own lists as well that may include the larger national list, as well as specific diseases to that state or territory. And on this list, it's in terms of the viruses I showed on that initial table, we're just talking about Japanese encephalitis and West Nile clinical disease. Now that's an interesting one in that it really is focusing on exotic strains of West Nile as opposed to the local Kunjun variant. So for mosquito surveillance, again, this is probably very similar to some of the mosquito programs that are active in the US. This is looking at long-term measurement of mosquito populations to allow the determination of things like baseline abundance. So what is normal, and using that to determine unusual activity. This can be used to identify mosquito species, population densities and age structure, and virus infection rates for early predictive data, together with environmental modelling. It's typically performed over the wet season and in summer and spring months here in Australia, which we're just going into. We're in spring already, in fact. And it also allows detection of virus from vectors and determination of which vectors carry certain viruses by that identification. Provides information about circulating arboviruses, including new and emerging viruses, and a number of laboratory techniques such as cell culture or PCR can be used. Where most labs are going these days that are doing this work is definitely in PCR, but also whole trap grinds using either PCR or next generation sequencing technologies. Sentinel animals are also used in Australia. This has usually been chicken sentinels, although pigs and cattle have also been used. Cattle are still used particularly for livestock diseases. And normally this involves year-round or seasonal serological surveillance to detect antibodies to the target arbovirus. And this can act as a very effective early warning system and typically triggers public health warnings and targeted surveillance. Such as the one you can see down here, the headline for Murray Valley encephalitis. And this was a sort of headline we were used to seeing before the emergence of JE. MVE was I guess one of the headline and high risk arboviruses that we look out for on a seasonal basis. So just to, I guess, summarise what the different state and territories do for their surveillance, and this is in operation before the emergence of JE, and I should say that all of the states and territories are now or have been revisiting and replanning their surveillance to take into account the emergence of JE. But prior to that, the focus was on medically important arboviruses of Australia, but particularly Murray Valley encephalitis, Kunjun, Ross River, Barmah Forest, and in Queensland and Northern Australia, dengue was a part of that mix as well. The focus was seasonal, mainly spring, summer, and opportunistic for mosquito trapping, and for sentinel chicken flocks, this was year-round. Or seasonal for some states. Other forms of surveillance are our first points of entry. This focuses on international airports and seaports, and the goal here is to prevent incursions of exotic mosquitoes at the border, and primarily involves vector monitoring at these ports in all of the states and territories, and involves mosquito trapping as well as DNA analysis. This is undertaken by our Federal Department of Agriculture, Fisheries and Forestries, and this has resulted in numerous detections of larvae in adults at airports. It's quite an effective surveillance system, and we often see ED species mosquitoes detected in this way. But to date, there have been no exotic mosquitoes that have established that we're aware of in breeding colonies, in breeding populations. With the exception of Aculex tridinarincus that I'll discuss later on. Another long-standing surveillance program that's been running has been run by the Department of Agriculture, and this is one called the Northern Australian Quarantine. The Northern Australian Quarantine Strategy. That shouldn't be Inspection Service. Otherwise known as NARCS, and this provides an early warning system for introduction of exotic pests, weeds and disease across Northern Australia. So you can see in the figure at the top, this covers a vast range of border or land in Australia's north. And it's delivered via a network of scientific and operational staff based at various sites along this border that you can see. And for JEV, what this involves is targeted sampling of feral pigs and livestock. And a good example of that is what gets done in the Northern Peninsula area at the very northern tip. I'll just change my pointer so I can show you things. The very northern tip of Queensland is known as the Cape York Peninsula, and at the very top of that is the Northern Peninsula area. And this is a focus area for the NARCS program. And this is a year-round surveillance program, and it's also expanded and intensified in response to cases or outbreaks such as the JE outbreaks in 1995 and to 2021 and 2022. Frequent JE activity has been detected via this program in this northern area of northern Queensland and the Torres Straits, which separate Australia's mainland from Papua New Guinea here. The Torres Straits, I should say, is a part of Australia, and there have been frequent detections in these islands that you can see there, as well as in the Northern Peninsula area. And often, I should say, just to describe some of these pictures that you can see at the bottom right of the screen, the surveillance will involve aerial culling of animals, followed by necropsy and sampling that you can see there. Sentinel pig herds have also been used, and mainly in the Torres Strait Islands, and that began in 1995 following the JE outbreak there as part of the NARCS program, and involves serological testing of young pigs, relatively small herds, 3 to 8 animals, and this was focused on the wet season. Involved antibody and virus testing, and there were sentinels located throughout the Torres Strait Islands and the Northern Peninsula area. This was gradually disbanded from 2006, and the last remaining herd was employed in the 2011-12 wet season. And it was considered that maintaining amplifier hosts as dedicated sentinels was a public health risk, but maintaining this sort of sentinel herd was also costly and operationally challenging in a remote location. So let's turn to the current outbreak then. I know you've already heard from Dr. Shipp and Dr. Richardson and Dr. Gleeson last night about the Australian outbreak. I'll hopefully have a point of difference to tell you about, but just to, I guess, reintroduce you to the outbreak that we've experienced this year. The first reports of reproductive disease came in late February in Queensland, New South Wales, Victoria, and South Australia. And of these, the earliest onset reported was from mid to late January in southern New South Wales. We've had over 80 infected piggeries now spread throughout the states. The two most affected states have been New South Wales and Victoria, that have accounted for around 3/4 of those infected piggeries, but also significant numbers in Queensland and South Australia. I guess what has been an interesting development has been the testing or sampling and testing of feral pigs in Northern Australia, and what has been found so far is active infections in over 50 feral pigs tested by PCR in 6 local government areas of the Northern Territory. And this is surveillance that has been performed by NARCS. We've been involved in this in terms of laboratory testing, along with the Berrima Veterinary Laboratory in Northern Territory. And just to take a diagnostic diversion at this point, for this surveillance, tonsils proved to be a really valuable specimen type. So we found this was, when we did get tonsils from infected pigs, they were almost always positive. And this supports what we know about replication of the tonsils and persistence of the virus in that tissue during infection from experimental studies. And one of the first to report on that was the Ricklin paper in Nature Communications, which some of you may be familiar with. Moving on to human cases, this is the summary table for the cases and deaths in parentheses for 2022. You may have seen this yesterday. In total, 41 cases and 3 fatalities across the 4 states affected. The cases were based on, confirmed cases rather, were based on laboratory definitive evidence based on the lab case definition, while probable cases were laboratory suggestive evidence, but without a rule out of related viruses. There are also cases in Northern Territory in 2021, and I'll talk about those in a little bit later. Other animal cases that we found, well, there were several suspected horse cases which broadly fit the probable definition for humans in terms of the lab case definition. And interestingly, we saw a single alpaca case in the Adelaide Plains of South Australia in March, and this was the first known occurrence of JE, and this is a neurological disease case in alpacas. But I guess it's analogous to what has been seen occasionally or rarely in alpacas infected with West Nile in the US, where you are. So it's interesting to see this analogous infection for JE in a new species during this sort of large explosive outbreak, which we sometimes see for arboviruses in this situation. Okay, looking at the distribution now, hopefully I'm not doubling up on what you saw yesterday, but this is the map taken from the World Organization for Animal Health, or WOA, WAHA's system. This was taken from the final follow-up report, number 4, which was going back to April, but it still has a lot of the detail. There's 3/4 of the cases that of the infected properties shown here, and the ones that are missing broadly fit into this distribution. So it's still quite a relevant figure. And you can see that the cases are, for the most part, west of the Great Dividing Range, which runs down this western part. You can see the elevated regions, the darker regions here, going from Queensland all the way down New South Wales and into Victoria. And from there, what we see is most of the cases are associated with water bodies or water courses, river systems, such as the Murray-Darling running along here, and then into South Australia. The alpaca cases here in the orange, green triangle rather, and these orange circles are single pig infected properties. And on the right is an inset just to show you a more detailed fine map of where these cases exist. For some time it was thought there was a northern southern divide along this area here in Victoria, but since then we've found serological evidence further south, so it's not as clear cut as we I guess we're hoping. Now, when we overlay that map with the publicly available data for human and mosquito detections, we see a similar distribution of those. And of that publicly available data, you can see here the New South Wales human cases and mosquito detections, and Sentinel chicken detections as well. So in terms of mosquitoes from New South Wales, there were detections in, so I'm just going to try and move the video. My head is blocking the text. I've fixed that now. Mosquito collections in southern New South Wales between the 10th and 24th of January in Victoria in several locations. There was also a single antibody positive Sentinel chicken from Deniliquin, and that's down here in the south of New South Wales, which was interesting in that despite that Sentinel chicken system that I described earlier being present, it was only able to detect a single chicken, which suggests that that system wasn't quite calibrated for detecting JE. So for outbreak response, in terms of surveillance, mainly I won't talk about control actually. What this involved was retrospective or expanded testing of mosquito and animal samples. This included or involved state and territory arbovirus control programs and the mosquito and sentinel chicken programs that I mentioned before. So samples that have been collected previously for identifying MV or Kunjun or Ross River and Bummer Forest were hauled out of the freezers and retested for JE. We also looked at prior cases of animal disease, particularly reproductive disease in pigs that hadn't been tested for JE. And this also included, the retrospective testing also included samples that had been collected for the NARCS program as part of the National Northern Surveillance Program. There was also targeted mosquito surveillance in affected areas and piggeries, and alongside that there was a National Animal Health Surveillance Plan that was drawn up. that involved a limiting surveillance of domestic pigs, as well as a CSIRO survey of selected infected properties to look at within herd transmission dynamics. And in terms of mosquito control, this primarily included insecticide treatment and source reduction at breeding sites and piggeries. And I believe Dr. Richards talked about mosquito control on piggeries yesterday, so I'll leave that there. So what do we find from the retrospective testing? From serological testing from samples collected in the Northern Territory, we found Sentinel cattle sampled in November 2020 and in March and April 2021. So well before the 2022 outbreak, we found serological evidence. We also found antibodies in a healthy feral pig from the Coburg Peninsula in the Northern Territory. That's just up here as part of the NARC survey. And one of the, I guess, really interesting detections that made a splash at the time was a positive detection in a retrospective case of reproductive disease in a piggery in northern Queensland that dated back to April 2021. So this lineage had clearly been circulating well before the large outbreak in Northern Australia, which was a little disturbing, I guess, once we realised that. We all realised, we'd been living with this virus for many months before it caused the outbreak. So just to take a diagnostic detour, if I may, we've been heavily involved in doing diagnostics at ACDP for both national surveillance programs and for state and territory diagnostics. We've also done some work with public health laboratories. Occasionally we get involved in human case investigations. We'll also do mosquito confirmatory testing. And I guess where we started out was in the laboratory diagnosis of the index case going back to the 25th of February. This was from a Queensland piggery. It was on a Friday. We got our positive case, and we worked through the weekend to get the whole genome by Monday the following week. And once we got that, we distributed that genome to the public health networks for both animal and human health. And that's, I guess, that was really the start of things, the start of a very busy year, I can tell you. So in terms of the species we tested, you can see there pigs, horses, mosquitoes, alpaca, chickens, and cattle. Of those, pigs have by far been the predominant sample and submission that we've received. So far we've received 473 submissions. In fact, that's higher now. This is a little out of date, and just under 6,000 samples. Of those, the majority have been for exclusion and confirmatory testing, but we've also received significant numbers of samples for surveillance, and we're well over 7,000 samples for surveillance now, and also just a small number of samples for health certification. And I should say we used different algorithms for exclusion testing and disease investigation versus surveillance testing. And I'd be happy to discuss those further after the talk. I won't go into them now. Just something I wanted to emphasize. Kirsty mentioned to me that there was a question last night on our PCR capacity. So one of the things that we found early on was that many of the PCR tests and assays used in Australia by different laboratories weren't particularly sensitive at detecting this novel genotype 4 strain. And we were very fortunate in being in a position that prior to the outbreak, ACDP was using a test capable of detecting all 5 of the genotypes of JE. And that's something we updated about 3 years ago through involvement in a project in Papua New Guinea, where we needed to make sure the tests we were using were going to target all of the genotypes, because we didn't really know what was circulating there. So I look back at that project, and I really thank our lucky stars that we reviewed our test at that stage, because it really put us in a good position for this year. But that the same couldn't be said for many other labs who were using older assays that were designed based on limited sequencing data that were fine for picking up genotypes 1, 2, and 3, but not genotypes 4 and 5. And to exemplify this, there are a couple of examples on the right-hand side. One is the Tiwi Islands JE case from 2021. This was work that we were involved in with PathWest, and this paper by Sakazwa and others will hopefully be published soon, and you'll be able to read that. And you can see here that these are brain specimens from this case using an older test at PathWest and the new test that they used based on the Baruka et al. Study, which worked very well. We used the Shao et al. Study, and you can see the comparison for pig sample testing with our previous assay that we were using, and it was really night and day in both cases. Just a quick note on flavivirology. I've got the heading here, science or witchcraft. I guess that's a question that's come up a number of times in a number of the working groups that I've been in that have included veterinary epidemiologists and clinicians who really, I guess, don't like flavivirus serology. It's not clear cut. You rarely get a yes, no answer or positive, negative. And there's usually interpretation of a number of different formats of testing to work through to get an answer. So as many of you already will know, flaviviruses are classified as serogroups based on their antigenic relatedness. And in Australia, the JEV serogroup viruses are JE, MV, and West Nile, which is a very similar situation, I think, to the US serogroup viruses, where you're really just substituting St. Louis encephalitis in for MV. So I'd expect should JE emerge in the US, and hopefully it won't, but this issue of serological cross-reactivity is something that you will probably need to deal with. So when you have viruses belong to the same serogroup co-circulating, then you need to test in parallel to assess the levels of cross-reacting antibody. Primary infections can generally be diagnosed using serology, but not always. And this is typically based on either a single reaction in the panel of tests that you're using, or that can be identified based on a fourfold or greater level of antibody titer for the specific virus, infecting virus. Now, following secondary infection, things get more complicated, particularly with a related virus, when we typically see a broadening of the antibody response. Highest levels of antibody are normally to the primary infecting virus through an anamnestic response. And in Australia, interpretation may just not be possible in older animals from hyperendemic regions, such as in Northern Australia, where these viruses are all circulating and animals are being repeatedly infected. So we typically see just broad cross-reactions for older feral pigs in Northern Australia, but when we look at so domestic pigs in Southern Australia, that's when we can start to see better discrimination in the antibody responses. Maternal antibody in piglets can also mask natural exposure, as can the vaccine responses as well. Okay, so I guess this outbreak in Australia raised a number of questions very early on, and many of these we're still grappling with, and that's, I guess, what I'd like to finish on for the last part of the talk. It was obviously an unprecedented outbreak in Southeast Australia, and the sort of questions we had are, where did it come from? What are the major vectors? What are the major vertebrate hosts other than pigs? It was quite clear early on that pigs were a major amplifier. And a question where, I guess, thinking very seriously about now and preparing for is, will JE return to Southeast Australia this coming spring, summer? And has it become endemic? And I guess that last question most people are thinking, myself included, is that yes, particularly in Northern Australia, but perhaps not in Southeastern Australia. And these, I guess, thinking about these questions brings to mind questions that we had back in the 90s when JE emerged in the Torres Straits in the in 1995. And just to show this, I guess this is a trip down memory lane for me. This is a 2014 slide that I put together for a conference in Nepal, of all places, around why JE has not become exotic in Northern Australia. And keep in mind, Torres Straits is, I've lost my pointer, where are we? I've lost my pointer, sorry, but Torres Straits is up to the north of that sort of red coloured tip of Queensland. And really that's where JE had stayed for many, many years. And the question was, why didn't it spread further south? And there are lots of, some hypotheses around that. And, you know, why didn't that happen when we had all the ingredients sitting there? And we still don't really know the answer of that question. But it's interesting that that question has now been flipped. So to, I guess, take a step back, what I'd like to do is provide a very brief history of JE in Australia. So I mentioned this Torres Strait outbreak in 1995. This involved 3 human cases, 1 fatality. But there were antibodies detected widely in the resident human population and also in pigs that were located on the Torres Straits. In 1998, there was the first mainland human case in the Mitchell River in that the area that you can see marked in orange there. I've got my pointer back. That was on the Western Cape York Peninsula. There were virus isolations from pigs in 1998, also from Culex and Alarostris mosquitoes in 95 and 98, and from a mixed Culexidians subgroup collection from 2004. And these Australian viruses were over 99% identical to Papua New Guinea isolates that were made from the mid to late 90s, providing a very strong evidence that PNG was the origin of that activity. And I mean, looking at the map, it makes a lot of sense as well. So fast forward 20 or so years, and we saw this unusual, unexpected, fatal human case on the Tiwi Islands in Northern Territory. So this is the Tiwi Islands here. Darwin is this population centre that you can see here, and Tiwi Islands just sit off the north of the Northern Territory. This was a locally acquired case. The person hadn't moved or travelled prior to becoming infected, and it's estimated that the infection was probably acquired towards the end of January in 2021. And as I said before, this was the real surprise was that it was a genotype 4. There was a second case in the Northern Territory identified this year retrospectively after additional testing was done, and this occurred just a few months after the Tiwi Island case in May 2021, and fortunately that person recovered. So in terms of the question, what are the mechanisms of spread of JE to Australia? So waterbirds were implicated in the spread from PNG in the late 90s. There are several artiodavien species known to move between Australia and PNG, such as herons, egrets, and bitterns. And they have sufficient connectivity across waterbird populations within Australia, moving from north to south, to allow that to happen via mosquito vectors. Microbats and fruit bats have also been proposed as potential vector hosts capable of introducing JE into Northern Australia, and there's experimental evidence that you may be aware of for Torres electo to transmit experimentally infected Torres electo to transmit virus to feeding mosquitoes, and JE has also been detected in a wide range of bat species in Indonesia. And bats may also play a role as a maintenance host. Windblown mosquitoes were also proposed as a mechanism of transfer of JE from PNG or even Timor as well. And to support this, there was this identification of Culex tridinorhynchus discovered for the first time in Northern Territory from collections in Darwin and the Catherine further south in February and May 2020. And this was identified or shown to be identical or virtually identical to a haplotype collected in Timor-Leste in 1999, providing pretty good evidence that there's an introduction pathway between Timor-Leste and the Northern Territory. The authors thought this was most likely to be windborne spread, but we also couldn't rule out airborne or aircraft or shipping. Now, some work that Peter Durr at ACDP and Karen Graham in his group have been working on is wind dispersal modelling using this system, this modelling system called TAPIS that Peter and Karen developed, which is a big data modelling tool for assessing pest and pathogen airborne spread. And they performed these analyses after the outbreak this year to look at the potential introductory pathways, and they looked at Timor-Leste, which I'll show first, and a number of different dispersion sites over every single day across the summer period in 2020, 2021, and 2022. These 3 days here are around when we, I guess, speculate that the Tiwi Island case acquired the infection. You can see that there's significant wind dispersion going right across the northern parts of Northern Territory and the Tiwi Islands, which correlates very nicely with that case. Now, looking at PNG, in February of that year, we also saw wind dispersion events going into Queensland as well, and this precedes the detection of the reproductive case in April 2021 in around the same area. Now, I'm just mindful of the time. I might just make this my last slide. In terms of environmental and ecological factors, in Australia in 2022, we've had a La Nina, a southern oscillation dominant for the past 2 years, which has led to wetter conditions in eastern Australia. It's also the dominant cycle this year for this spring summer, and you can see in the map on the top right that shows rainfall. This was very much above average, or above average for most of Australia. Leading into last summer. And this led to flooding of waterways and lakes in inland Australia, providing a breeding ground for mosquitoes, and also these waterways attracted water birds. There was also several species of potential mosquito vectors present in these areas, and several waterbird breeding sites were established west of the Great Divide and along the Murray-Darling Basin. From Queensland into South Australia, all of these were active last season and overlap with rural areas, including where pigs are present, and not to forget the large feral pig population that spreads across northern and southeast Australia, which you can see in a somewhat outdated map, but still roughly accurate on the bottom right. Just to, I guess, highlight what we're already seeing, we're already seeing flooding in Australia. So this was a photo taken of the Barwon River in Geelong. This is at the end of my street. This was on Saturday night. This river's about 300 metres from me, and this was at 3.86 metres, the 5th highest on record level, and it flooded the surrounding roads and walkways. It's going down now, fortunately. So to turn to potential vectors and vertebrates now, the question we had there, and looking at our JE amplification cycle, in terms of vectors, there are several species in Australia that could act as a vector for JE. Top of the list is Culex rostris, mainly because of the fact that it's been associated with detections in the past. It's also a primary vector of Murray Valley encephalitis, but as you can see, there's a number of other species there that are either vectors of JE in other countries or are minor vectors regionally that also circulate in Australia. Now, in terms of water birds, this is another major question we have, and still have some way to answer this. There are a number of species, potential species that may act as reservoir hosts, such as the Nankeen night heron and plumed egret, both of which are also important water bird hosts for Murray Valley encephalitis and West Nile. But there are also other species like the little egret, white-faced heron, and magpie geese that have populations within Australasia. Ducks have also been discussed as a potential reservoir host, and in particular, a question is there for the role of ducklings, and it's been found experimentally that ducklings can generate a viremia high enough to infect biting mosquitoes, but that level of viremia drops away as the ducks get older. in terms of their ability to support it the older they get. And just to finish on this section, marsupials have also come up. Australia obviously has a large population and different species of marsupials. At ACDP many years ago, this is going back up to just after the first outbreaks, there were experimental infections done of a couple of different kangaroo species, as well as brush-tailed possums. And of these possums were the only animals to show a significant level of viremia following experimental infection, which lasted several days. We didn't have the capacity back then to do mosquito infections, but even so, the level of viremia that was seen was considered to be high enough to support potential transmission. And there are lots of these possums in residential areas as well. All right, so to finish up on then, as we approach the next wet season and spring, summer, we, I guess, are faced with a number of questions, a number of challenges, existing questions that I've just discussed. If JE has become endemic, which is one of our questions, a continued and strengthened One Health approach will be needed for expanded surveillance and disease mitigation strategies. Expanded and enhanced surveillance may benefit from novel approaches. Some of these that we've considered include oral fluids for pen-based surveillance of piggeries. Effluent testing for piggeries has also been considered, and both of these activities are likely to commence in the coming months as pilot studies initially in different states. And consideration of other sentinel animal systems has also been made, particularly dogs, goats, and cattle that have been used in other countries as potentially sensitive alternative sentinel systems. It's also recognised that mosquito surveillance should be boosted, especially near piggeries and effluent ponds, but also near or within waterbird habitats. And one of the major gaps at the moment is waterbird surveillance to identify important reservoir hosts. And that's something that we hope will happen coming into the next season. And with that, I'll leave it there and thank you for listening. Thank you for your attendance and happy to take any questions.
David, thank you very much. So questions from the audience, please. Eric, please, yes.
We have a virtual question, a question from a virtual attendee, Jerome Kortikos from Boehringer Ingelheim, says, very nice presentation, thank you. Can you explain why it took more than 6 months before a full genome sequence of the outbreak strains was submitted to GenBank?
Yeah, gosh, that's a great question to start with. Thank you, Jerome. So that came from us, from ACDP, and it was the pig genome sequence, I believe. So in Australia, there are a lot of sensitivities, particularly around pig genome data, not so much the sequence, but the metadata. So we had to work through, I guess, the various permissions and approvals and get the relevant decision makers comfortable with the idea of putting that sequence into a publicly available database. And that took some time. It was also in the context of, a raging outbreak as well, for which there were many other competing priorities. I should say, though, that once we sequenced that genome, we distributed it to our internal public health laboratory network. So the Australian laboratories that needed that sequence had access to it. And we also provided it on request to ACDP as well. And I should also say it took a number of weeks once we submitted it to GenBank before it was released. So it was probably more like 4 months after we sequenced it that we submitted it. So apologies it took so long, but it was just one of those, I guess, frustrating things for us, but challenging things to work through.
David, thank you, Jerome. Thank you very much for that question, Megan.
Yes. Hi, David. This is Megan Niedermorter from the Swine Health Information Center. Thank you so much for being a part of this symposium and your presentation. I had 2 questions. One is based on the PCR detection of the virus. And you mentioned tonsil from feral pigs as well as neurologic tissue. I'm curious what sample type you are recommending for commercial piggeries, such as when they submit mummified fetuses, is it a homogenate, or are there other sample types where you have recommended for submission and detection of the virus? And then my second question is with regards to the surveillance. Australia obviously has fantastic surveillance programs, and the US is not currently doing any surveillance. And so I'm curious where you see the most value, and it perhaps is speculation, but what's the most value with regards to the surveillance programs? Is it mosquito, feral swine, commercial swine, and based on your experience?
Thanks, Megan. Thanks for those great questions. To address the first one, tissue types, for aborted fetuses and neonates that euthanized or die early on after birth, CNS tissue was probably the most sensitive tissue. We pretty much always got quite low CT samples back from that for PCR. That said, I think were able to detect JE in just about any of the tissues that we got from mummified or aborted fetuses. So the virus seemed to be replicating throughout the bodies, but CNS was, I think, well, I know was a more sensitive sample. Just as I guess a curiosity around that, we also tried isolation, virus isolation. We did a lot of virus isolation attempts, and we weren't able to isolate from that CNS tissue, even when we had CT values in the teens, which really surprised us and initially. We were working with our pathologist at ACDP, and Willie Suen here made the observation that those CNS tissues were, for the most part, highly degraded and probably been, as you know, I guess, sitting in the placenta, gradually decaying, and there was a lot of autolysis around those samples that probably explained why we couldn't isolate viable virus back. However, the genome seemed to be protected. For feral pigs, tonsils were best. So I guess that was a key learning from our experience of sampling feral animals or feral pigs. We could also detect in whole blood from those animals, but when we compared or when we matched whole blood versus tonsils from individual pigs, we more often detected in tonsils than blood. I hope that answers that question. In terms of where I see the most value in surveillance systems, that's a really good question, and it's one we grapple with here as well. And you know, my reply to this may well be different to the next person's, but I guess mosquito surveillance is considered by many as being a high value activity. It used to be something that was labor intensive. It often involved virus isolation. The standard was to use small pools of speciated mosquitoes, but where things are moving now is to use whole traps and do whole trap grinds without the speciating. And that allows a much more rapid turnaround of mosquito trapping, and it's all molecular based. That doesn't mean you can't go back and do isolation and that sort of thing. It does mean your speciation is obviously compromised, but there are potential ways around that using next-gen sequencing technologies as well in bioinformatics. Yeah, I might leave that there. Obviously, antibody surveillance is, I think, quite useful, and chicken surveillance has been really the mainstay of Australian arbovirus surveillance for decades and has worked very well for MV and Kunjin. I guess we've got questions about its effectiveness for JE, and we haven't quite worked that one out yet. So I guess watch this space.
David, thank you very much. Stephen, yes.
Hey, David, Steve Reckham from USDA, this One Health. Just a quick follow-up on the mosquito surveillance. You said that at the ports, it's supported by the federal ag system, I believe. But is that the case across the rest of the country, or is that left to the states and territories? How does it work outside of the ports?
So for those international ports, regardless of the state or territory, that's run by the Federal Department of Agriculture.
And how about surveillance outside of the ports?
Oh, sorry, I misheard you. Surveillance outside of the ports is state and territory based. It's operationalised by relevant state and territory, either public health or environmental health units, and so they each are funded separately through the states and operate in slightly different ways, but broadly similar.
Thank you, David, very much. Are there other questions from the audience? Eric, yes.
Just one more question from Jerome Corticas. Are there any plans to perform vector competence studies involving pigs? Are lab-reared Culex annulorostris available for such studies?
There are, so we're doing vector competence studies at the moment, not using pigs, just using artificial feeding systems, blood feeding systems. We have a laboratory colony of Culex annulorostris, which is a southern Australian colony, which may well behave, well, hopefully not too different, but there may be subtle differences with northern populations of annulorostris. So we're hoping to finish that initial study by about the end of November. We don't have plans yet of doing pig studies, but you know, I guess that's an interesting question. I guess we'll get through the first round of experiments first, and then see where to go from there. I believe there are other laboratories in Australia that are looking at different populations or laboratory reared populations of annulorostris as well, but I'm not quite sure where they're up to.
Thank you, David. Other questions from the audience? Yes, please, Harry.
Yeah, hi, David. Harry Snelson with the American Association of Swine Veterinarians. Just curious, are sheep not susceptible?
No, I don't think so. I'm not aware of any sheep cases of JE. Please correct me, anyone, if you are aware of that. If they are, they're very rare if they have occurred. There have been some rare cases in cattle. I think there's maybe 2 publications on that. And West Nile can also cause sheep cases as well. So, you know, it wouldn't be surprising if there is the occasional rare case of JE, but to my knowledge and expectation, they wouldn't be a major vertebrate host.
Thank you very much, David. Other questions from the audience? Eric, are there any additional questions online? So, David, thank you very much. It was a fascinating presentation. It's very good to see you again.